Hi, I'm Justin Balsor. I'm a graduate student in the Minds Neuroscience program at McMaster University. I work in the Visual Neuroscience lab headed by Dr. Kathy Murphy. My research focuses on characterizing sets of synaptic proteins in both human and animal visual cortex across the lifespan. In order to look at these synaptic proteins, I had to improve upon an existing method for procuring and enriching the synaptic proteasome, which is known as the synaptoneurosome preparation. I was able to do this by taking advantage of modern homogenization and filtration techniques. Of great help for that was the FastPrep 24 benchtop homogenizer. In order to examine these synaptic proteins, it's very helpful to enrich your tissues by putting them through a synaptoneurosome preparation. The FastPrep 24 was really crucial for our improvement on the existing protocol. The old homogenization technique required you to put your tissue samples in a glass Downs homogenizer, and we found a lot of variability between experimenters in our lab, some people being able to homogenize much better than others. With the FastPrep 24, we noticed, of course, you can just put your samples inside individual tubes, load up to 24 samples into the machine at once, and have, again, up to 24 samples fully, completely, and evenly homogenized in less than 40 seconds. Because the FastPrep 24 is able to homogenize up to 24 samples at once for you, and because it does such a great job of evenly homogenizing your samples across the board, we're able to procure higher synaptic protein yields, and it's also a very easy protocol to convey to a new scientist or to convey in a journal publication. The old homogenization techniques were often very qualitative when describing this protocol, often saying things like 10 to 12 turns, applying even pressure, whereas now when I have to teach, say, a new student in the lab how to do tissue homogenizations, it's a matter of telling them at what speed and for how long to run the FastPrep machine. Because of the updated biosafety regulations that we have in Canada, the FastPrep is just great because you load all of your samples inside of contained environments inside each of the lysing matrix tubes, and then you further enclose them inside of the dome before homogenizing. This allows us to perform our entire protocol inside of a fume hood where it is safe and enclosed and we're protected from any viruses that may be in the tissues. This is very important because our research focuses on both monkey and human tissue as well as other animal species across the lifespan. So our valuable tissue is completely enclosed and contained, and we're sure that we get as much tissue and protein yield from our samples as possible. So this tissue prep that we've developed is going to be great for my research, as it will allow us to look at these very special proteins and enrich for them, and we can examine things such as the role that these proteins play in cortical circuits, especially in the visual cortex, and use that information to determine how these proteins play a role in visual development. Thank you for taking the time to listen to my research and for how FastPrep was able to play an important role in furthering my education.